Thanks everybody for coming today. It's my pleasure to introduce today's speaker, Dr. Gisela Camacho Hernandez. Do you include the Andrea?
That's fine.
Dr. Camacho Hernandez is coming to us from the National Institute on Drug Abuse, which is part of the National Institutes of Health. It's part of the intramural research program in the National Institutes of Health. So the NIH, you might, if you're familiar with it at all, it's probably because they fund grants to universities like ours and researchers like ours, but that's the extramural program. So that's that reviews and sends out that reviews grants and sends out that money. The intramural research program is lots of high-level research scientists doing really fantastic cutting edge stuff, but they don't have to apply for grants the same way that we do. So a lot of those labs are located in Bethesda on the main campus, but NIDA, National Institute on Drug Abuse, is actually located in Baltimore, along with the National Institute on Aging. I've known Dr. Camacho Hernandez for a few years now. She is a member of the Amy Hawk Newman lab at NIDA IRP, which is a lab I did my postdoctoral research. So I was the only non-chemist in that lab. So she's going to talk a lot more about the chemistry side of things, the medicinal chemistry side of things, developing new medications. I had the distinct pleasure of working with all those chemists who created all those compounds, and I got to test them. So Dr. Camacho Hernandez did her BS in biopharmaceutical chemistry at Autonomous University of Baja California. She then got an MS in biopharmaceutical chemistry, also at Autonomous University of Baja California, and did a PhD in pharmacology. That was through a joint program from the Autonomous University of Baja California and UC San Diego. And what year did you join November 2018. Okay, so just before everything fell apart. So, let's all welcome Dr. Camacho Hernandez.
Thank you, Tom, for the kind introduction. And it's really my pleasure. I really want to thank Tom and Greg for inviting me to speak to you about the project that I've been working on during my postdoctoral years at the Newman Lab. So today I'll be talking about attempts to illuminating the monoamine transporter by developing fluorescently labeled ligands to study the dopamine and the norepinephrine transporter. That's the so, I was going to talk a little bit about myself, to introduce myself actually, although Tom already introduced me. It's fine. So I was very recently from California, Mexico. I actually did my yes in biopharmaceutical chemistry in the Autonomous University of Baja California, and my master's degree was on the same institution. Then I had the opportunity to join a combined program between my Autonomous University of Baja California and UCSD, which granted me the opportunity to do my research experience and training at this institution, which was a fantastic experience for me. And I'll tell you a bit of the story after the presentation, but I basically commuted the 4 years of PhD from Mexico to La Jolla every day. So for all of those that are commuters, I feel you, I see you, I didn't. And in November 2019, I actually decided to move from coast. So I came to the East Coast and joined the National Institute on Drug Abuse to do my postdoctoral work. As Tom mentioned, the NIDA IRP is located in Baltimore, Maryland, so we're very close. We have excellent research going on that you can actually check on the web page. We have also postdoctoral programs. We have the postdoctoral programs, summer program. So if you guys are interested, you can check it out. Our mission is to conduct state-of-the-art basic preclinical clinical research on mechanisms that underlie substance use disorder, addiction, and relapse. Ultimately, we hope that these efforts will grant us new methods for the prevention and treatment of substance use disorders. So because of this, the Newman Lab, which I am part of, has been focusing on a lot of targets associated with substance use disorders. And one of them is monoamine transporters. So the transporters are transmembrane proteins, and they are comprised of 3 members. dopamine transporter, the serotonin transporter, and the norepinephrine transporter, which I'm going to be referring as the SERT and NET during my talk. And the main function is to mediate the re-uptake of previously released monoamines from the extracellular space into the cell. And this process is usually sodium and chloride permitted. So it is.
We are actually interested in this strategy because there's dysregulation in neuropsychiatric disorders, including substance use disorders. So there are several techniques, and we have been working in the Newman lab developing not only compounds as potential therapeutics, but also chemical probes. And these chemical probes, in general, we can divide them to visualize and to actually study the distribution of this membrane proteins. And we can divide this visualization process broadly into the genetic and non-genetic approach. A genetic approach can be a protein that it's fused to YFP, GFP, for instance. And a non-genetic approach can be a small molecule. And of course, as a chemist, this is where I actually got very interested on. Today, I will be talking to you about fluorescently labeled ligands. And these compounds actually comprise of three parts: the ligand, a linker, and a fluorescent dye. So an important advantage versus the genetic approach is that we can use these compounds to actually study proteins that are endogenously expressed, meaning that we can actually tag neurons, for instance, or tissue. And how this system works is this is a reversible system. It's basically you have your protein of interest, then you apply your compound, then the ligand will actually bind, usually in the orthosteric binding pocket. You excite the fluorescent dye, and then you get, a beautiful image, or you can actually detect this by flow cytometry, etc. So one of the first compounds that the Newman lab developed is this JHC1064, and was developed almost 20 years ago. And to the best of our knowledge, is the first compound that was actually used to label the dopamine transporter in neurons. And this compound actually, it's quite wonderful and has been our superstar in the lab for many, many years and has been used for many groups. And one of the characteristics is that it actually binds to the three transporters quite similarly. You can see in here that I will be talking about the Ki value, which is basically a value that will talk about the affinity of the compound for, in this case, either the dopamine, the serotonin, or the norepinephrine transporter. Just a reference, the lower the value, the higher the affinity. But another important characteristic of this compound is that it actually has a very slow off rate. So you will remember in my previous slide, I showed you that it was a reversible process, so meaning that eventually that compound should dissociate. Well, this compound actually loves to be on the binding pocket of the transporter so much that it just really never dissociates, even though it's not really covalently bound. And because of this characteristic, our longtime collaborators from the University of Copenhagen has been using it to study internalization processes of the three transporters. And one of the cool things about this compound is actually reveal that the internalization process of that, when it was expressed in an heterologous system like HEC293 cells, compared to when it was expressed endogenously, was different, highlighting why it's so important to actually study proteins in their native environment. Another interesting purpose of this compound that was also done by our collaborators in Copenhagen is that they utilize our compound to assess the different expression between this DAT mutant, seen here, and that wild type. This mutation is highly prevalent in early on stage Parkinson's. So you can see that we can use these compounds like very broadly in tissue or in a heterologous system as well. So I talked at the beginning of my talk that it comprised of three features, right? The fluorescent dye, the linker, and the parent ligand. But I haven't really talked about what each part of this molecule does. So overall, all of your molecule, all of the structure, it's going to influence how it binds to the binding pocket. But that being said, the parent ligand plays a fundamental role, meaning that if the parent ligand has a higher affinity, it's likely that you're going to get a good affinity probe. Likely, not a rule. If it's not selective, it's likely that your probe, it's not going to be selective as well. So as an example, this JHC1064, it's a cocaine-based analog. So the parent ligand is this MFC212, and here we see the binding affinity. So one of the things that you can see is that once we introduce the linker and this bulky dye, it actually lost affinity. So it's very important for us as a medicinal chemist to actually understand what part of the molecule we can actually touch. We can actually introduce a linker, because this can really affect completely the binding to a point that it's just not going to bind anymore. And we do that through structure-activity relationships. Another important characteristic is the fluorescent dye that we chose. And this is because it always depends, where is your protein of interest? Is it in the membrane? Then you can pick a fluorescent dye that is cellular unpermeable. Is it inside the membrane? Then you have to choose a fluorescent dye that will have the ability to actually cross the membrane. So thankfully, right now, we have such a big toolbox of fluorescent dyes available, and most of them are commercially available, actually, that can give us, from confocal microscopy study to actually super resolution microscopy, we can actually see the dye very close to what the target is. This is kind of like very nice. And as an example, we actually generated a few years ago an analog of JHC164 that the only difference between this one and MFC918 is the dye. So in this case, we have Oregon green, instead of the rhodamine red. This dye is much more hydrophilic. And because of that, we were able to actually utilize it to label dopamine accents in rhesus macaques tissue. We couldn't use JHC164, although we tried, because rhodamine red tends to be much more lipophilic, so it was giving us a lot of non-specific labeling. So even in that case, when we design, or when we are actually trying to choose what probe to use to study what we want to study, we need to be mindful also on the properties of the probe. So we also have switched rhodamine red for something that it's much more suitable for super resolution microscopy. So you can see that by switching the actual dye, we don't really lose so much the binding affinity. We went from 17 nanomolar in dopamine transporter to 6.3. But with this compound, DG380, that would also play a little bit with the linker. And in this particular case, is because this dye tends to be quite lipophilic, we decided to add the PEG linker. We can not only use it to do classic confocal microscopy, but we can also use it to do these storm studies, which is much more super resolution microscopy experiments. So another thing that we have explored is that I have shown you compounds that basically bind with very high affinity to the 3 monoamine transporters, but I haven't shown you any selected compounds. So what will it happen if we want to study a system where all of them are together or 2 of them are together? So we cannot really use JHC164 because then we will not be really labeling one of them. We were going to be labeling everything, right? So I mentioned to you when I was showing the different components of the fluorescently labeled ligands that the parent ligand really plays an important role in selectivity. So what we did in here to generate a SERT preferential binder was actually to switch the cocaine-based parent ligand for citalopram, which is a known serotonin re-uptake inhibitor that has very high affinity for the serotonin transporter. And you can see that we generated an analog, basically just switching this portion of the structure, but now it's preferential and selective for SERT. So this worked out very well. But at this point, we were still not having anything for NET, and we didn't have anything for DAT. So that's when I joined the lab. And this project started actually with Andrea Casiraghi, who was a visiting graduate student from the University of Vienna. And what we did in here is kind of like use the same rationale and try to switch this parent ligand with something much more preferential for NET. So our criteria to choose a parent ligand is something that has high binding affinity. And I mentioned this is very important because since we're going to introduce this linker and fluorescent dye, we know that binding affinity is going to go down. High selectivity because we want something selective for NET. And structure activity relationships available. So this SAR was already in the literature, and it was important for us because it told us where in the molecule we can actually add, or we can try to explore adding the linker and the fluorescent dye. And here in isoxetine, I'm pointing out with the error in these two sites that we explored in california. So they're dancing a little bit. And now the chemistry. So I'm a medicinal chemist. I need to show at least a couple of slides showing chemistry. No worries, I'm not going to go into the details of every single reaction that I made, but I try to point out in each step what was the modification so you can kind of follow it. There is a rationale behind how we design the synthetic scheme. So it's not like we go to the lab and we just put all that together. We have to think about it, and I will tell you why. So as you can see in here, we started with this 3-bromobenzylaldehyde, and then after 3 steps, we generated basically the core of the parent ligand. So I have isoxetine up here for you as a reference. Then we started working in the what I call the handle, which is basically the position where we're going to introduce the linker of the fluorescent dye. And by an aromatic cyanation, we introduce an additional carbon that subsequently this nitrile is reduced to generate this primeramine that it's going to react with a rhodamine NHS ester that already has the linker. So the reason why we designed this this synthetic scheme in this way to actually put the fluorescent dye at the end is because a couple of reasons. One, the fluorescent dyes tends to be quite pricey. So we usually get them in 5 milligrams, 10 milligrams. And you're in your last step, so you're doing a reaction of 5, 10 milligrams. If you have ever done synthetic organic chemistry after doing or getting 5 milligrams of product, the last thing that you want to do is another step. That's one thing. The other thing is that dyes can photobleach very easily. So the conditions of the chemistry also get very tricky. So you need to be very mindful and careful of that. But the other reason, and the third one, is that this synthetic scheme allows us to be quite versatile in the sense that if we generate a lot of this intermediate, then we can add whatever fluorescent dye that we want. So in this case, we're adding rhodamine red, but if we need the Oregon green, we can do it. If we need the Janelia fluorescent dye, we can do it. If we want side 3, we can do it. So it gives us a lot of flexibility. All right, so after we get our compounds, we make sure that they're good, that they are what we think they are, they're characterized, they're pure. What do we want to do? We want to see how introducing the linker and the fluorescent dye actually affects the binding affinity, right? So we perform radioligand binding assay, and this is how we get this Ki values that I have been talking about. So this is the isoxetine-based compound that I just introduced you in the first synthetic scheme that I showed you. And as you can see, the binding for NET is quite good. It's 43 nanomolar, and it's quite selective with respect to that and SERT, especially if we compare it to JHC164 that I have here by reference. So we're quite excited about this compound. And you may think, okay, that sounds easy. You just switch the parent ligand and you get whatever you want. Not really. So we tried with talopram in 2 areas based on SAR. And as you can see, none of our probes actually bind to NET. So we were quite disappointed at this point, but at least we had one time. So it's not a very straightforward process. We still need to do the chemistry. We still need to kind of make the compounds and see whether they're gonna work or not. But at least at this point, we had this one. So we decided that, okay, maybe it's time to see whether this compound actually labels the NET. So we sent this compound all the way to Vienna, to the Medical University of Vienna, actually, where Dino and Deborah performed confocal microscopy studies. What they have is a cell line expressing YFP NET, and this is very important because it serves as control. We can see that NET is there. And then they added a 10 nanomolar concentration of our compound. So what they observed is that at 0 minutes and then 1 minute, and then you can see that the rhodamine signal starts to increase until it saturates at 20 minutes. So now we can see the rhodamine signal in the membrane of this cell. And you can get very excited and say, okay, this compound is working. But at this point, we don't really know if it's actually labeling NET or if it's just labeling the membrane or if it's non-specific labeling. So in order to assess this, what Deborah and Dino did was introducing a high concentration of isoxetine to basically just block all the NET binding, and then adding the 10 nanomolar concentration that we did in the previous experiments that I showed to you. Et voila, we don't see any labeling in the membrane. So at this point, I can summarize the first part of my talk, which is that we have developed an isoxetine-based fluorescent probe, AC01146, with high affinity for NET, moderate affinity for SERT, and low binding affinity for that, in radioligand competitive binding assay employing rat brain tissue. So all of these experiments were done actually in rat brain tissue. Unfortunately, even though we tried in 2 different positions, none of the talopram compounds were active at NET, demonstrating that the 2 substitutions that actually we choose, that they just not tolerate any chemistry or any bulkiness around it. And our same collaborators and then actually already utilized this compound in a following study to study actually the PIP2 role on NET oligomerization. So let's go back to our toolbox. So our toolbox now, we have JHC164, which is the superstar, but its MAT's not selective. We have DG380, who is very good for super resolution microscopy study, but again, it's not selective. We have now a SERT preferential binder. Sorry, and we have a NET preferential binder. But I mentioned at the beginning that there's 3 monoamine transporters, so we're missing the dopamine transporter preferential ligand. So when we were wrapping up the NET project, I talked to my advisor and I said, you know, I'm curious to see whether we can use some of our own molecules that we make in-house to see if we can get something that is preferential for that. And she told me, okay, go for it. But okay. So I utilized the same rationale, but instead of using the norepinephrine reuptake inhibitors, I utilized this atypical DAT inhibitor. So the great advantage is that we actually developed these compounds for medication for cocaine use disorders in the lab. So we know the chemistry very well. We know where to actually touch the molecule. So that was kind of quite what really pushed me to actually follow up with this project. So again, same criteria, high affinity. You can see both of them have high affinity. Selectivity, these 2 compounds basically bind in the micromolar range to SERT, and if they don't bind to NET. And lastly, again, we know the chemistry, so we knew where exactly, and I'm highlighting here with arrows, where we actually place or we look for to add the linker and the dye. And they're dancing again because if the NET did, why they cannot. So chemistry again. A little bit of chemistry that I'm showing you. And in here, the only thing that I want to highlight is that after 3 steps, we can generate this intermediate. And if you see the intermediate, it's quite similar to JHC 089 and 087. And it just gives me access to basically all of the compounds that I'm going to be talking about and that I will show you. So I can also generate this in the gram scale. But although the chemistry seems quite simple, I needed another 9 different schemes to actually get all the compounds that I'm going to show you today. But for sake of time and sake of everyone, I'm not going to show all the chemistry that we did, but you can find it in this paper that we just published. And if you have questions, please shoot me an email or come talk to me. Okay, so I work alongside a postdoc for 2 years, Amarachi Okaram. Had a blast with her. Now she's in med school in Mount Sinai, and she helped me to actually test our compounds in, again, radioligand binding assay. And these are the first 2 compounds I'm showing you are based on JHC 8089. And you can see that once we introduce the linker and the dye, the binding affinity for that went down, although they're quite selective with respect to SERT and NET. And the difference between these 2 structures is just this OH that is present in this compound, which seems that it doesn't really affect the binding for that. Then I made a compound to kind of like see whether pushing the linker and the fluorescent dye, where I think it's the binding pocket, because this is a transporter. So I thought, okay, maybe this compound goes inside and binds, but if the linker is too short and the dye is too short, maybe it's just not allowing it to bind so nicely. So maybe if I push it out of the binding pocket, the affinity can go much better. Well, it It did to some extent. So now we have 236. But what it was very interesting of this compound is that I actually start interacting now with MET. So we lost the selectivity. So you see, sometimes you modify and you fix something, but then you go and actually, you know, make things worse on another side. So it's kind of like a dance in the lab at the end of the day. But what about JHC 8087? So this is our best compound for that so far, G0304. And we explored two sites on this aromatic ring. In this case is the ortho position, and you can see it has a 140 nanomolar affinity for that, and it's quite selective over SERT and NET. Try to explore the same thing with the same rationale to kind of like increase the linker. In this case, it actually did not really benefit the binding affinity, and I saw the same thing I see in this previous compounds. We actually lost selectivity and started to interacting with MET as well, which we thought was kind of interesting. Okay, so these two are placed on the ortho position, but what about the para position? Well, it was very well tolerated, and you can see in here, the binding affinity is quite similar to GC0304, and it really retains binding affinity, basically doesn't bind to NET. Okay, the next thing that I wanted to test is to see how versatile this scaffold can be. Meaning, I have now rhodamine red, but what would happen if I actually switch it for a different dye? So what I did was basically switch in the last step. That's why the versatility of the synthetic scheme is so important. I switched it for the sulfoside 5. And as you can see, we were super excited because it's quite versatile. The binding affinity for the dopamine transporter and rat brain tissue, it's basically the same as this compound. So we can, this basically what it's telling us is that we can play around with the dyes depending on the type of experiments that you want to do, or if you want to introduce a bunch of different colors, you can do that as well. So like, okay, this is very exciting. So this compound, I tested also in the human dopamine transporter, and you see it's this very similar affinity that is the rat, and super selective as well. Okay, so I put this, that is an advertisement to the International Transmembrane Transport Society, but more as a highlighting why it was so important to attend conference meetings. And this actually conference made a big difference because I was presenting my data, I was showing my compounds, and I had the pleasure to meet Dr. Kushboe, who is a long-time collaborator of my advisor, Amy Newman, and Aditya Gopinath, who at that moment was a PhD student. And he came to my poster, I started talking about all these dyes, and he got super excited, and he told me, you know what, I'm developing this flow cytometry method. I have developed it, but I would like to try one of your compounds. The only thing that I need is a compound that doesn't cross the membrane. And I told him, okay, I got you. I know which compound I can give you. So what Dr. Kushboe and Aditya are doing in their lab in the University of Gainesville, it's actually trying to investigate the brain to immune system communication. So they are basically, they're focusing primarily on Parkinson's. So what they do is try to understand how the dysregulation of that in the brain affects immunity response. And they have, again, focusing a lot on Parkinson's, but they have been doing a lot of work also in substance use disorder. So Aditya developed this beautiful method on flow cytometry where he can collect blood, in this case with human blood samples. Then he isolates monocytes. And then the monocytes, he treats them by several steps of permeation with an antibody. Follow the tagging of this DAT, who is actually tagged to a fluorescent dye. He can detect the levels of that in these monocytes. And his method works very well. It's very replicable and you know, it's very nice. The only problem is that this antibody binds to the intracellular loop of the dopamine transporter. So they have to do a lot of permeation experiments in the steps, in the process. So just to prepare the sample, it takes him more than 2.5 hours. So that's why he was interested to see whether a dye that actually binds in the surface will actually help them to remove all these extra steps that he had to do. So as Dr. Kushboe has been a long time collaborator of Dr. Newman, they have been used in other studies, JHC 164. they say, okay, let's try this compound and let's see if it works. So you can see that they did confocal microscopy studies, and the compound was labeling very beautifully. They ran their controls and flow cytometry. So in here, this is with the DAT antibody. So you see he detects 23%. But no matter the concentration that he was using of JHC 164, 10, 100, 1000 nanomolar, he was not detecting any DAT with this compound. Now, another downside of this is that it also binds to NET, and based on their studies, they know that NET is also present in the monocytes. So that's why he got very interested on my compounds, because he said, okay, they're DAT selective, they don't bind to that, this will be perfect. So I was talking to him during the meeting, he told me that when he was preparing the samples is that there was the color was getting lower as the time progressed of incubation. So he said, I don't know if the diet's actually permeating, which is possible for JHC 164, or if it's photo bleaching. I don't know exactly what is happening, but I think if we try something that it doesn't permeate, it might work. So I gave him GC0438, which has this disulfoside 5, and it's in the literature has been reported that it doesn't cross the membrane. It's actually quite hydrophilic, this compound. So then what he did was basically running parallel his experiments with the antibody and with our compound. He got the human blood samples, isolated the monocytes, and then did the treatment with the antibody and with my compound. And then he actually get the detection via flow cytometry. So this is the antibody. Again, the sample preparation, it's 165 minutes. And then he utilized GC0438. And as you can see, it's basically the same. So at 1 micromolar, he was able to detect exactly the same thing that he was detecting with the antibody. The phenomenal thing is that now, instead of having to prepare his sample for 165 minutes, we went down to 30. So he was quite excited because now we know that this compound binds to the membrane, so he doesn't really need to do all this permeation steps. So he was like, okay, I saved a lot of time, this is great. But let's see how versatile this compound is through different species. So in their studies, what they're trying to do, they have access to collect blood sample from patients in different stages of Parkinson's. But then they want to translate what is actually happening and try to understand it through an animal model. So they do kind of like a reverse translation strategy. So they use a lot of mice. So he wanted to see whether this compound actually had the same ability to label that in wild type mouse monocytes. So he collected blood, same treatment, get the monocytes, and then utilize the antibody and GC0436. So you can see he detects the antibody 29%. And then when he utilized my compound, he sees exactly the same. So he was very excited. He said, I can use the same compound for both the animal and human samples. But just to be extra cautious, they have actually a DAT knockout mouse. So in theory, if you have a DAT knockout, you shouldn't be able to detect any DAT, right? So they collected blood, isolated the monocytes, did all the treatment with the antibody in our compound. And as you can see, like in the antibody, we don't see any detection with that. And this really assured us that whatever he was getting was because the compound was actually labeling that. So our compound is quite selective. So a summary of second part, we have developed modafinil-like, and I say modafinil-like because JHC 8, 087, and 089 were based on modafinil, although they have much higher affinity for than modafinil, so like in 2 micromolar. With decreased selectivity over that in radioligand competitive binding assay employing rat brain tissue, GC0438 successfully labeled HDAT and NDAT on circulating peripheral blood monocytes, reducing the time of sample preparation from 165 minutes to 30 minutes. And I'm quite excited to share this with all of you because we got the supplementary cover of Jack's Gold. So this is an open access journal. So if you want to know more details, you can check it out. This is just fresh out of the oven, basically. We just got this out. And currently Aditya, he still works in the University of Gainesville. So they're currently using our compound still to assess the role of that in peripheral immune cells and the association with Parkinson's disease. And with that, I would like to thank the Newman Lab, of course, my mentor, Dr. Amy Newman, and all the amazing people that work in the lab, who I had a blast. People that have been involved directly, but you know, it's always very nice to be in a nice environment because there's always bad days in the lab. So having a nice place that you can go and talk around, it's always very nice and reassuring. Of course, our collaborators from the University of Milan, from the Medical University of Vienna, the Vienna University of Technology, and of course, Aditya and Dr. Prashwali of the University of Florida in Gainesville. Thank you again to Tom and Greg for having me. It has been a pleasure to be here, and I'll be very happy to answer any questions if I can. Thank you.
Anybody has any questions back, a little back? So maybe ask the first one.
So you use the you've done the competition assay in order to determine selectivity of the net binding. And that was with the YFP net. Can you do co-localization studies as well for the two fluorescent reporters between the YFP net and your red, I'm guessing, signal on your initial fluorophore?
I think we can do that, yes. I haven't done these experiments, but I think it's possible.
This is super cool work. I'm curious, the difference in substitution position and length of linker effect on selectivity was a little surprising to me. I would expect there to be an effect. I wouldn't necessarily expect it to be that big. And I'm curious if you have a sense of why. Like, is it a function of the flexibility of the molecule overall? Are there any changes to the spectral properties that can give you an indication of that?
I think that the three monoamine transporters share so much sequence similarity, especially DAT and NET, for instance. So that, although they are overridden in their endogenous neurotransmitter, actually that can also transport NET, for instance. So my thought, and this is only speculation, okay, because we haven't done any computation, we don't really know what's going on outside. My thought is that when it starts to actually interacting more outside the binding pocket, there can be residues that are much more present in all of the three proteins that can actually help to actually get much more binding to that. They don't get, if you see, it's not that they went from 10 or not binding to 100 nanomolar. They started interacting more slightly a little bit, but they were still in the micromolar range. So that's why I think there has to be much more interaction of the linker. That's why I always said that yes, the pyruvate is very important, but at the end of the day, you have like an overall big structure that it's not like, you just have the linker and the fluorescent dye and it's just outside doing nothing. It's actually can fold and interact with the whole protein outside as well. So I think it has to do much more with this type of interactions that might be available in the three proteins, right, than anything else. That's my thought.
That makes sense. Have you guys, like, I know in fluorescence microscopy, you sort of get single wavelength kinds of answers. Like you excite with a specific wavelength and you're looking for a specific wavelength. So any changes in the spectral properties of the fluorophore are sort of invisible by that method. Has anybody done any sort of more detailed and careful analytical fluorescence spectroscopy to understand those behaviors?
No, we have not. It would be a very good idea actually to understand more, but no, we haven't done that. We haven't done that.
Okay, thanks. Students? No.
This is a very good question. In this particular case, we don't have agonists and antagonists because this is a transporter. So we have like substrates or inhibitors, right? So we know in the literature and many work about actually some collaborators of us that the transport actually can be blocked by a very simple modification, just adding maybe a methyl group, for instance. So in this particular case, we know that I mean, we haven't run into any difference, but we know that once we introduce the linker and the fluorescent domain, this is likely to be an inhibitor because the protein will not be able to actually transport it in. Actually, if it can transport it in, that will be super cool, but we haven't found the way to actually add such bulky components and be still transported into the system. Now, this is the transporters. Now, if we think about it with a receptor that we actually have the agonist and antagonist, then it will matter in a sense of what is the experiment, what are the studies that you want to do? Because an agonist, if it binds, it will trigger a response, right? So for it, if you just want to label, it might not matter. If you're actually doing some functional experiments and you have an agonist that actually triggers a response, it might give you some results different than if you actually use an inhibitor. So you have to more or less think about what type of experiments that you're going to perform in order to say, well, we're going to use an inhibitor or we're going to use an agonist if you're talking more about a receptor, if that makes sense.
Yes, thank you.
Okay, I'll start asking questions. Oh, wait, no.
It will not overlap, but yeah, for sure, it will matter if you have like the reserve and all of this to actually be transported in. I don't know, to be honest, the answer to that.
I know a little bit.
Okay, go for it.
So the level of transport also relates to the likelihood that the transporters get phosphorylated. And that in turn relates to the likelihood that the transporter stays on the surface membrane. And so it's not necessarily direct one-to-one always as well, but if it's not actively transporting, it's more likely to lose its phosphorylation or get phosphorylated and thus change its trafficking. Okay. Yes. Yes, yeah, exactly.
In that way. So it's more than being transported in and actually gets trapped and it goes all the way with the protein.
In reference to the Parkinson's overlap.
Is there a way that you could study disease progression through that method of measuring the transporters?
So what I know that BPS do, and I don't know if they're doing exactly with our compound, but they were actually, one of the papers actually that I showed, they were checking the levels of that in patients that were just diagnosed with Parkinson's that have not taken any medication versus patients that were already taking medication versus patients that had been longer time, and see whether those levels were actually leveling up, which it did actually. So they saw different levels in what they call like early stages without any medication. And then once they take medication, it seems like they start the expression in the immune cells, they start leveling up to normal patients, I would say, or patients that were healthy. So it seems like it can.
Thank you.
This might be a really dumb question.
So
Why don't your molecules bind to like dopamine receptors? Why are they binding only to the transporters instead of to the GPCRs that also bind dopamine? These proteins that recognize the same molecule, right? So do you see residual affinity for the GPCRs?
all these compounds, they haven't tested on the dopamine receptors, but the JHCA, the parent ligands, they actually don't bind. They don't bind to the dopamine receptors. I think they have been tested at least in D2, D3, and D4 in my understanding, and they don't bind. And yes, that is true what you said, that if it binds to the transporter similarly, but it's not dopamine, so it structurally is different. Now, this is also a inhibitor. It's not exactly a substrate. So the structurally is different, where it binds is different, the conformation that actually stabilizes is different. So there's differences between one and the other. But yeah, as I mentioned, the fluorescent case, we haven't tested in the receptor, but the parent ligands, we know that they don't bind to the receptor.
Thanks.
When it internalizes, BC receptor internalizing, your diet is also internalized. In early endosomes, degrading.
My understanding is this is a paper that was done previously by our collaborators in Copenhagen, but they do follow the actually I will say the endosomes. Yes, I will say the endosome, although I'm not 100% sure, but they can actually follow it.
In regard to the leader, that actually will dictate how far down into the transport channel, so that there's a broader perspective of deeper he's going, and he's halfway in between, so it's like the other side, or they, also the complex with the bottom, upper bottom, section, so where are they?
Oh my God, I would love to have like a solid answer to this, and that is something that I have been like asking you because can we do any computation now? Because we work very closely to a computational person in NIDA that he has done a lot of studies with the parent compound, so they have done a lot to know whether they actually were located, if they are atypical inhibitors, if they're typical. So they haven't done a lot of work on like, can you just put these compounds to see? We haven't done it. So my answer to this in terms of speculation is purely based on the parent compound and where it binds. So it actually sits kind of like in the middle. If it is, in this case, I don't know if it's an atypical or typical because they go toward either the inward facing conformation or the outward facing conformation, but I have no idea for this particular compounds.
Yeah, I was interested in the pattern.
Sure.
We're facing oddly one of the as an analog of so atypical.
So, and we have right now, we're finishing a paper where we think that the sulfoxide, because it has a sulfite instead of a sulfoxide compared to modafinil. We think, or what we're seeing in computational studies is that sulfoxides, it's not a rule, but they are more likely to be atypical. These ones have sulfite, so they are more likely to be typical. But again, we haven't done the study, we don't really know for like good data.
Possible to add like a calculating way sort of towards the fluorescent end, so that you could then potentially permanently label the transporter.
That's a great question. So there was a postdoc before me that was working, trying to make what we call cleavable probes. So the system from this, it works differently because I showed you at the beginning of the presentation that this is a reversible, in theory, reversible process if your compound actually dissociates eventually. With the cleavable probes, what it happens is that it goes, it binds, and it has a site that will react with residues around it and will form a new covalent bond. So then the compound that actually took it there will wash out, and then you have now permanently labeled your protein of interest. And he tried to open with the transporter. He never was able to get any compound at one. I can say that I went to the sites project because it was quite challenging. But I had was working with the mu-opioid receptor, and I managed to get a compound actually that labels, and we have some preliminary data already that labels the mu-opioid receptor, and it's a cleavable probe, and it's working. So far, it's working very, very nicely, but that's for the receptor. The transporter, our collaborators think that the problem is that compared to the receptor, it seems like the receptor, you have a component that binds much more in the periphery. The transporter, it goes much more deeper. So what they think is that when it goes deeper, it gets very tricky, very complicated. So yeah, he tried for many years, but he was not able to get it on and try it. So yeah, but that would be very cool if we can get it.
So for the students in the audience, one thing I'd let you make sure you know, so tomorrow, there is a career panel, and Dr. Camacho Hernandez is part of that panel. It's all about biomedical careers in the workforce and what a PhD can do for you, you know. So please plan to attend that. That's at 12:30 tomorrow in Business Hall. And there's a networking event after that's over, so you can actually chat with Dr. Concha Hernandez and the other panelists and learn more. Now is a perfectly reasonable time for you to ask questions about career-related stuff if you would like. If you're too shy, you could come up right after we're done here and ask directly. Okay, I think we're done.
Get nervous and I start.
Just adding more things. I say too much.
Nice to meet you as well.